طيب بسم الله الرحمن الرحيم. احد السيشنز اللي ريليتد للسي اي اي كامبريدج how organisms are classified. فبعضكم طلب الكلاسيفيكيشن بتاع الاورجانيزمز. هو موضوع بسيط وسهل جدا و applied in our real life، فسهل جدا ان احنا نعرفه. هو مش موجود في الادكسل، بس موجود في الكامبريدج، فما فيش مشكله. طيب ايه المطلوب؟ الاوبجكتيفز بتاعتنا ان احنا نعرف how classification system aim to reflect evolutionary relationship. يعني انه classification system بيوضح نظريه التطور بتاع داروين وايه relationship between different kingdoms, and explains that classification is traditionally based on studies in morphology and anatomy. Mainly, the classification of organisms are based on morphology, the external features of different organisms, and the anatomy, the structure of their body, and explains that the sequence of bases of DNA and the amino acids in protein are used as a more accurate way for classification nowadays. With recent advances in science, the sequences of bases of DNA is nowadays used to classify organisms instead of depending only on external morphology, which is subjective. Subjective يعني depends on someone point of view, and anatomy, and this is objective but less accurate also. So the sequence of base of DNA is recently introduced to be used as a way, as a mean to classify organisms, and explains that organisms which share more recent ancestor are more closely related, have base sequences of DNA that are more similar than those that share only a distant ancestor. So how to classify? First of all, the process of classification depend on this scientist in 1970 or 60. Linnaeus established this system for classification of organisms. He classified organisms into kingdom, phylum, class, order, family, genus, and species. And all living organisms are classified according to this. Okay, according to Linnaeus system. So this Linnaeus is a Swedish naturalist, scientist. In 1735, he started to put a classification to classify organisms into smaller and more specialized groups depending on their morphology, which is the overall form and shape of the organism, the external features, the apparent external features of the organisms, like whether they have legs, they have wings, they have hair, they have how many segments of their body, and so on. And the anatomy, the detailed body structures are determined by this section. In his binomial, and he named this system of classification as a binomial system of classification. Every living organism has a unique name composed of two parts. He named the organisms into names. Every organism is named by two parts. One first name is referring to the genus, and the second name is referring to the species. And he used the Latin language to write a name to name the organisms. And when he's writing this genus and the species, he's writing them in italic mode. So the genus always start by a capital letter, and the species in a small letter, uppercase letter. The first letter of the genus in naming of the microorganisms is uppercase letter, and the first letter of the species is a small case letter. And when typed binomial names are always italic. So if like this example, Homo sapien for human being. So H, Homo, H is capital, Homo is a genus, and sapien is the species. And the questions are coming in the exam, on the, especially in paper 2, in the multiple choice question. So he's mentioning, he's telling what is the first word homo is referring to, or he is asking in the opposite direction. So he's telling where is the genus here, where is the species here. So just remember that the first is the genus, the second is species, the genus starting by uppercase letter. Okay, for example, just to train yourself, this is a baby crab. This is a crab. So the photo is a photo of a crab, and the scientific name is the Xanthias lamaricae. So where is the genus? The first one is the genus, the second one is the species, and the Xanthias is starting by capital X, uppercase X. So this is the genus, this is the species. Very simple, and it depends on your observation. You have to be a good observer. And for your exam, the order for classification can be remembered by using this mnemonics. This is very famous. That King Philip came over for Grand's spaghetti. So King, if you take the first letter, capital letter of each word, then you will remember that the Linnaeus system of classification is kingdom, phylum, class, order, family, genus, and species. In the exam, no need to memorize all of this, because the exam is measuring to which extent you are understanding the system of classification. So we will ask questions that will measure your understanding and not your remember this. So the recent way for classification of the organisms are the DNA-based sequences. We will take the DNA later on, and you can see the video in the DNA synthesis. We have a video in the Telegram group and in the YouTube channel about the DNA. We will tell you that DNA is deoxyribonucleic acid, and it's composed of a backbone of sugar and the phosphate group and a nitrogenous base. We have 4 different nitrogenous bases. According to the base sequences, the DNA, and similarity between different species in the base sequence of DNA, the more similar the base sequence in the DNA, the more the species are related to each other. So this DNA-based sequence are used to tell that this species is related or not related, is near or far from the other species. As you know, as we will take later on, these base sequences are coding for formation of special amino acids. So according to the base sequences, the amino acid sequences will be, and a different type of protein will be produced by this amino acid sequences. So there will be similarity in the base sequences. They will go for similar amino acids in between species, and this will make similar protein between species. So both species will be more or less equal to each other. So if you wanted to know the species is similar to the other species, try to test for DNA-based sequence for the species number 1 and species number 2. If they are similar, then they are related to each other. Look at this question. The exam question is coming like this. This is very common in your exam. That this is a base sequence: adenosine, guanine, cytosine, thymine, and these are the 4 nitrogenous base in the DNA structure. This is the base sequences in this piece of DNA. So the question is asking for this section of DNA from an organism that's most CK chimpanzee. So which one? Which sequence of this is related to the same? This sequences of nitrogenous base is coming from the DNA of chimpanzee. So which one of this is related in the species to this chimpanzee? So look at this: A, G, C, T, A, C, A, G, A, T. So here is a difference only in one nitrogenous base, but in this, so A, G, C, number B, and the T, T, A, C, A, A, G, G, T, T. 2 are different here. 2 nitrogenous bases are different. Look at this: A, T, this one is different. C, A, C. So a lot of different here. This is not correct. ATC. So this is not correct also. So the first one, number A, is different only in one nitrogenous base. Number B is different in two nitrogenous base. So look, when you found that, so the proper answer is number A, because the base sequence is the same. And take care, don't fall in the trap of making a complementary base sequence. No, this is not complementary. It is similar base sequences, not a complementary base sequence. So if you are taking complementary base sequences, so we are not coding, we are not making a protein. Complementary is for making a protein from the DNA, but here the similar base sequences have to be similar as much as we can. So we will tell that this number A is mostly related to the same species like this chimpanzee. Okay. Okay. Another important part of this, according to your syllabus, is constructing and using the dichotomous key. The dichotomous key is a tool that allow user to decide or to identify a certain item, a tree, animal, simple object, and every object, either living or non-living in life, they are using the dichotomous key to decide which item we are dealing with. So it is not only related to biology, but it is related for life. So they are using for classifying chairs, they are using for classifying houses, they are using to classify trees, and so on. Any living or non-living thing can be used to be classified according to this dichotomous key. It is not only related to biology. So dichotomous means that it divides something into two parts. Therefore, it gives two choices, two options in each step. And then if you are following one option, this option will take you for another two options, and so on until you identify this object, either living or non-living, whatever. So the key is are mutually exclusive characteristics of biological organisms. So look at this photo. The first key is telling that leaf with several small leaflets. Is this diagram has several small leaflets? No, I think it has only one large leaf blade. So leaflets are broad and flat. So if you are choosing this number 2, leaf with one large leaf blade, then go for 3. So go directly for 3. Leaf with a smooth edge or leaf with a toothed edge. I think here it is with toothed edge, so we'll select the right answer is D. So like this, so you are following the starting with first key, and the first key will give you go to 3, go to 2, and then follow, and according to your observation and according to the drawing in front of you, you will select according to what you are seeing. Is this multiple leaflet or only one broad leaf? Then the next question has smooth or toothed edge. It has a toothed edge, so the correct answer is D, and so on. So for your exam, simple dichotomous almost always coming up in the multiple choice paper. Very occasionally they show up in the theory paper. Very rare to come the theory paper 4. So it will come paper 2, the multiple choice concept. When they do, and this question والسؤال ده مضمون جدا في الامتحان انه كلكم تاخدوا الدرجه بتاعته لانه على حسب observation and start with the first key they are giving it you and follow according to the diagram in front of you. And when they do, you almost always have to use one through serial information given for each step. Look at this question. Look at this question. Use the key to identify. Use the key to identify animal. Look at the diagram and use the key. First, has 2 wings, well, has 4 wings. 2 wings, well, 4 wings. Yes, excellent. 4 wings. So 4 wings, go for 3. 3 has feathery antenna or has smooth antenna. Feathery antenna and an antenna has projection, small projection, many projection. But this antenna are smooth. This is antenna, and antenna are smooth. So smooth antenna you will select. very easy, and marks are تعتبر مضمونة في الإجابة على هذا السؤال. The first division of things of classification هنبتدي ناخد بقى الفايف kingdoms اللي موجودين الانيمال plants fungi protists and prokaryotes. الفيرست فور بنسميهم يوكاريوتس واللاست للبروكاريوتس، وهناخد ان شاء الله في السيل ستراكشر هنعرف لو رجعت في الفيديو هتلاقينا عاملين كومباريزون بين البروكاريوتس واليوكاريوتس. وهناخد معلومات تفصيلية عن السيلولار ستراكشر of this animal, plant, fungi, and protists and prokaryotes. Here we'll take the external feature. We'll in Cambridge here is caring about the external feature of animal kingdom and the plant kingdom, fungi, and so on. Later on, we'll take the cellular structure of animal, plant, fungi, protists, and prokaryotes, viruses. So one of the best way to tell if an organism is an animal is to look at its cell under the microscope. Animal cell never have cell wall; it have cell membrane only. Cell wall is a unique feature of plant cell. Animal are classified into many phyla. Here are just some of these phyla. So animal kingdom are classified into vertebrates, annelids, molluscs, and nematodes, arthropods. And vertebrate will concentrate more on the differentiation between different vertebrates and different arthropods according to your syllabus. So we'll start directly. What's the meaning of vertebrate? Very common in the exam, because why you are calling them vertebrate? Because they have a backbone. There are 5 classes. في عندنا 5 classes تعدهم على إيدك. You need to be skilled in knowing the external feature. Here we are talking again, we are talking about the external features, how to differentiate between these 5 classes of vertebrates. And from the external features, not from under the microscope. External features, we are looking by naked eye on the appearance or the external features of the animal. What are the features you are seeing by your eye? To differentiate, to tell that this is amphibian, this is a bird, this is mammals, and so on. So in the exam tip, they are telling differentiate between visible features. So the questions coming clear visible feature, with giving you a picture or a photo or a diagram, and they tell you what are the visible features that tell that this is a bird. So from the diagram you have to tell. Don't tell from your information. That look at the diagram in front of you, and from this diagram tell them 1 or 2 or 3 according to the marks of the till the external features visible in front of you. And sometimes very rare the question will come for non-visible features. Then you will mention from your brain, from your mind, what are the non-visible features. Okay. Classifying vertebrates, they are called vertebrates because they have a backbone. And vertebrates are animal, related to animal kingdom. And we have 5 classes. We have mammals, fish, reptiles, amphibians, and birds. We'll take, first of all, I'd like to mention a key difference between some of them are called cold-blooded and warm-blooded. To the left are the cold-blooded, like frogs, crocodile, fish, and so on. It means that their temperature, their body temperature depends on the environmental temperature. But warm-blooded, they can control their internal temperature. They can control whatever the environmental temperature. But here the temperature on the left, the cold-blooded, the temperature of their body depends on the outside environmental temperature to the large extent. But to the right, this human being like chimpanzee, like dogs, birds, and cows, and different animals. So the different mammals, they are warm-blooded, means that they can control their internal temperature by themselves. So whatever the outside temperature, environmental temperature, they can maintain their internal temperature constant, whatever the changes in the external environment. So fish, as a first group related to mammals, the main characteristics of fish here, they have doors, they have fins. These fins help help them to move in water. They have a streamlined body covered with scales. They have scales. This feature is clear here. And the fish lays millions of tiny eggs, and fish breathe with their gills. The gills are here, and they are cold-blooded, so their temperature regulation depends on the outside environmental temperature. They swim with their fins. The fins help them to swim, to move in water, and their body is covered by scales, and they have gills that allow them to breathe for gas exchange, and they have a streamlined body covered with scales. The streamlined body helps them to move easily in water to decrease resistance to the flow of water. The second group is amphibians. Amphibians like frogs. Amphibians have moist skin. They have 4 legs. The external feature that you are seeing here, they have 4 legs, and they have air drums here. Moist skin is moist, and they breathe with gills when they are young, tadpoles, and when they are becoming older, they are using their lung. If they live on land and water, they can live on land and water, and they have 4 legs. They are cold-blooded, and their body temperature depends for a large extent on the environmental temperature. Reptiles, the third group is reptiles. Reptiles have dry skin, as you see, but amphibian has wet skins and scales. It has scaly skin, and some have legs, some has no legs. Like this crocodile has legs, like snakes has no legs, and they are cold-blooded also. Okay, and they are lay rubbery eggs. But here in amphibian, they have small gel-like eggs, lay small gel-like eggs. But amphibian reptiles have rubbery eggs. And the fourth group is birds. The main features they have beak, they have feather, and they have 2 legs, and they have 2 wings, and they are warm-blooded, so they can control their internal temperature, whatever the changes the external environmental temperature. They can fly, but in the exam they have feather, they have 2 wings, they have 2 legs, they have beak, and so on. They are putting hard-shelled eggs. hard-shelled eggs زي ما انتم بتشوفوا كده التشيكن eggs, hard shell, white hard shell eggs. But in case of reptiles, it is a rubbery eggs, and in case of amphibian, it is a small gel-like eggs. Okay. So mammals have hair and fur, hair and fur. Here there is a fur, and they are born alive and drink milk, are breastfeeding. And mammals breathe with their lungs. They have a special system for breathing and gas exchange. They are warm-blooded. Land mammals has 4 limbs, 2 arms and 2 legs, or 4 legs. They have air bone and a single jaw. They have single jaw here. And bats are the only mammals that can fly, and whales and dolphin can swim. Whales and dolphin are mammals also, because they're sharing the external features of mammals. The invertebrates, this organisms that animals that don't have a backbone. Some of them has legs, some has no legs. We will, according to your syllabus, you are concerned only with arthropods. They are the invertebrates that have jointed legs. They are classified into the following classes, but the main feature is that they have no backbone. They have exoskeleton, all of them. So if they can live in a dry place and not confined to water or moist place like most invertebrates. They have segmented body and jointed legs. We have 4 groups: the insect, arachnids, and crustaceans, and myriapods. And the insect, according to, we will differentiate between them according to the number of legs, according to the body segments, according to the presence or absence of antenna, and according to the wings, presence or absence of wings. So the first group, the insect, we have 6 legs. In arachnids, we have 8 legs, and in crustacean, we have 10 legs, and in myriapods, they have lots of legs. But take care. Sometimes in the exam, they are telling you 6 legs and or 3 pairs of legs. So if they are telling 3 pairs, yes, because it has 3 on this right side and left side, 3 on right and 3 on left. So these are called 3 pairs. 3 pairs, either you mention as 3 pairs or 6 legs. And here in arachnids also, you can mention 4 pairs of legs on each side of the body, or 4 pairs or 4 legs on each side of the body, or 4 pairs of legs in total, or 8 legs. So you either talk in the language of pairs, or don't mention pairs and mention the total number of legs. And it's better to mention the pairs. So this insect has 3 pairs, and arachnids has 4 pairs, and crustaceans has 5 pairs. All of them has exoskeleton and body parts like head, thorax, and abdomen. In insect, we have 3 body parts. In arachnids and crustacean, we have 2 body parts. Head and thorax. Head and chest. Thorax means chest. Head and chest are 1 part, and abdomen is another part. But in case of insects, they has head, thorax, and abdomen, 3 parts. Insect has 2 antennae. Arachnids has don't have antenna or wings, and crustaceans has 2 antennae, and myriapods has 2 antennae at the front, at the head. And about wings, antenna and wings, insect has have wings. Arachnids don't have antenna or wings, and the crustaceans, some of them has wings, some of them don't have wings. So myriapods don't have wings. About body segment, we are telling that the insect has 3 body parts or segment. Arachnids has 2 segments. Crustacean has 2 segments, and myriapods has a lot of segments. We will take, if you are looking in the exams, they will ask you for the external feature you are seeing. Just comment on the body parts, the presence of wings, and the presence of legs, presence of antenna. So 4 things, remember: body parts, antenna, wings, and number of legs. Okay. Accordingly, you can classify them into insect, arachnids, crustaceans, or myriapods. Students are often confused by different number of legs in insect, arachnids, and crustacean. So they often lose marks if you are starting that 3 legs and there are 3 pairs of legs. If you are talking about legs, mention that in insect we have 6 legs. In arachnids we have 8 legs, and so on. If you are talking about pairs of legs, so insect will have 3 pairs of legs, arachnids have 4 pairs of legs, and crustaceans has 5 pairs of legs. Okay, plant kingdom. Plants are multicellular organism, and cell wall is characteristically made of cellulose. They have cell wall outside the cell membrane. We'll take it in the cell structure in details. And this cell wall is made of cellulose, which is protecting and keeping the plant, keeping the cell, protecting the cell, and keeping the cell tough to afford the pressure inside the cell. They include mosses, ferns, and flowering plant. We have flowering and non-flowering plant, and plant classification in general. We have flowering. Flowering plant we are classifying into according to the seeds. We have the seeds has only 1 leaf called cotyledons, and we'll take this in details in the reproduction in plant chapter. So flowering plant are classified in 1 seed leaf. One seed has 1 leaf only. This leaf from which the embryo will grow and make the stem and root. And some flowering plant has 2 seed leaves, 2 cotyledons. We are calling this 2 cotyledon dicots. and the one seed leaf, which is one cotyledon, monocots. And the seeds that have two cotyledons, we are saying to die with two cotyledon, cotyledon. Cotyledon is a seed leaf. This is like the leaf present inside the seed from which the embryo will take their nutrition and start to grow when the circumstances is allowed. And plant, we have flowering and non-flowering plant. Non-flowering plant, we have to know that just to know that one of example of non-flowering plant is the furnace and the mosses. This is according to your syllabus. Only know like this non-flowering plant and a plant that has no flower. So one example of them is furnace and mosses. And the flowering plant, we are classifying them into according to the number of cotyledons. Cotyledons are a leaf present inside the seed from which the embryo will take its nutrition and growing up. When circumstance allowed. Circumstance means that availability of water and the availability temperature and light and soil and so on. So this according to the leaf inside the seed, which is the cotyledon. Either the flowering plant has one cotyledon or two cotyledons. If they have one cotyledon, they are calling them monocots. If they have two cotyledons, they are calling them dicots. So what this is a diagram showing the cotyledon, one cotyledon only. And here there are two cotyledons, and here is the growing embryo inside, and so on. Here is cotyledon, and the embryo starts to make radicals and the stem, and the first leaf starts to appear here. And here is the dicotyledon. The seed has two cotyledon, one right and one left. And then the plant will start growing from here, making the first leaf, and then to make stem and radicle and so on. So the flowering plant, they are sharing that they have roots, stem, and leaves, and they have xylem and phloem, and they reproduce by producing the seeds, and seeds are produced inside the ovary, inside the flower, and asexual reproduction is possible. We'll take all of this. So we are dividing the flowering plant into monocotyledons. They have only one cotyledon only. What are the cotyledons? They are the seed leaf. This has small leaf present inside the seed from which the embryo are taking nutrition and start to grow when circumstances allow. So this is cotyledon. Some flowering plant has one cotyledon only. They are calling it mono. It means one cotyledon, monocots. Or dicotyledon, يعني it has two cotyledon. They are calling it dicots. Okay, so what are the difference between monocotyledon and the dicotyledon? Monocots and the dicots. Monocots, the seed has single cotyledon, and the dicots, from the name, the seed has two cotyledon. What is the cotyledon? They are the seed leaf. Leaves, when the plant growing up, the leaves has parallel veins. The veins are parallel, but here the veins are branched inside. And the flower parts often in multiple of three, and you found the flower parts in multiple of three, like this 1, 2, 3, 1, 2, 3. The petals and stamen. Petals and stamen are in the 3 petals or 3 stamen, and so on. But here flower parts are often multiple of 5, 4, 5. And this is very common in the exam, in your exam, to differentiate between monocots and dicots. And they're telling why is this the monocots, why it's the dicots, and or multiple choice questions. Many are coming on this. But flower part in monocots, there are multiple of 3. So you found them 6 petals or stamen, or 9 petals or stamen, or 12 petals or stamen, a multiple of 3. But flower parts often are multiple of 4 or 5 in case of dicots. A vascular stem, if you are taking a cross section of the stem, you will find the vascular part scattered. The phloem and xylem is scattered in the cross section, bearing scattered. But in case of dicots, the vascular bundle, phloem and xylem are arranged into rings, as you are seeing here. So sometimes you're coming to the exam and telling you which one is dicots, which one is monocots. If they are scattered, the xylem and the phloem are scattered inside the cross section of stem, they are monocots. If they are arranged into rings, they are dicots. And roots, here you will find the roots also. This is sometimes coming in the exam to differentiate between monocots and dicots. So roots here are fibrous, and roots are hairy roots. But root here is tape root. Tape root is here, one root only, and going directly to the soil. But here you not found one main root, but you found a lot of roots, fibrous roots are there. So here is the difference between the leaves. Leaves are veins are parallel, and here leaves are veins are branched. This is dicots. In monocots, the floral parts of the flower are even multiple of three. But in flowers, in dicots, there are multiple parts, are multiple of 4 or 5. Vascular bundles are scattered in the cross section of the stem. Vascular bundles are arranged in a circular manner in a cross section of the stem. The roots in monocots are fibrous, and the roots in dicots are single main root originating from 8 small fibers. Tape root, we are calling this main root the tape root. Fungi, which is the third kingdom. Quickly, we have multicellular fungi. Some fungi are multicellular, has many cells, and some fungi are single cell, like yeast. Some fungi are pathogens, example, and this is coming in your exam. What are the diseases caused by the fungi? Mention one disease caused by fungi. We will mention that athlete's foot. They will found white patches like this under the fingers, under the toes, especially the little toe. Okay. This is a fungal infection. And the fungal structure, we have a multicellular fungus showing rounded spores, and these spores are carried in a thread-like. Fungal cell has a cell wall. If you are looking at the fungal cell under the microscope, you found that they have cell wall-like plant. But cell wall here, in case of fungus, as we've taken the chapter of cell structure, are made of chitin. And the plant cell wall is made of cellulose. So this is a difference between fungal cell and the plant cell. The multicellular fungi usually are arranged in something called a mycelium, which is made of thread-like structure called the hyphae. This thread-like structure called the hyphae, and this hyphae has many nuclei, and there is a spores carried on this hyphae, and so on. This is a multicellular fungi. Fungal nutrition, the fungi can't carry out photosynthesis. Instead, they are saprophytic. They are using saprophytic nutrition. Saprophytic nutrition, it means that they secrete enzyme into their food, so they will cause digestion of this food outside the fungal cell, and then they absorb the digested organic product. They may store carbohydrate as glycogen. Remember that plants store carbohydrate as starch. This is the second difference between fungal cell and plant cell. And we take this in the cell structure chapter. So bacterial cell. Bacteria are a small structure, usually monocellular. They don't have membrane-bound nuclei, and the genetic material are free in the cytoplasm like this. Large circular DNA molecule, and they have a small DNA, circle of DNA, they are calling it plasmid, and they have a cytoplasm and cell membrane and cell wall. And they have cell wall and cytoplasm containing glycogen granules, no nucleus, only DNA in the form of single coiled chromosome. DNA is very simple coiled chromosome, and they have a slim capsule, and sometimes some of them have flagella. Adaptation in bacterial cell to each function, and we'll take all of this in cell structure in details. And the DNA is found loose in the cytoplasm. It's called chromosomal DNA, and is not contained within the nucleus. There is no nucleus here. Plasmid, bacteria also have a small closed circle DNA called plasmid present in the cytoplasm. Plasmid can move from bacteria to another and giving a variation to another one, can move from one bacteria to another one, giving a variation, and it has a uses and application in the genetic engineering, as we'll take later on. Flagella, this is a singular flagellum. They can rotate or move to help the bacteria to move in the environment. Cell wall, bacterial cell wall provides structure and protection. Prototistics are single cell organisms. Some prototistics, such as amoeba, have feature like animal cell. Others, such as chlorella, have a chloroplast and are more like plant. This is very common in our life chlorella is very common, and this is a photo for chlorella. It has many chloroplasts, and it is able to do photosynthesis. Examples for pathogenic protistics: Plasmodium, the organisms that are causing malaria, and viruses. They are the last kingdom, and they are not a living organism. Example of viruses are influenza virus and HIV virus. And the virus structure is only a capsule, and this capsule is surrounding a genetic material, and it is not a living organism, because viruses by itself can't do any of the characteristics of the living organism. Virus can't move by itself. Virus can't reproduce. It can't grow. It can't excrete, and so on. So virus need to be introduced into a cell, and he is directing the nucleus of this living cell to do copies of its genetic material, and so on. So the simple structure of the virus is a capsule surrounding a genetic material. This genetic material may be a DNA or RNA, deoxyribonucleic acid, or ribonucleic acid, but not both of them. Either the virus will have genetic material of DNA or RNA. Example of the virus: influenza virus, HIV virus, and COVID-19, the SARS-CoV-2 virus. Okay, thank you, and we'll meet uh, in the next session. Till that, best of wishes for you. Thank you.